Welcome to Blood Vessels Part 2. Now that we finished discussing the anatomy of blood vessels, we're going to look at the physiology. How blood circulates through, how we control blood pressure. So the first term we'll need to know is blood flow. So blood flow is the amount of blood flowing through a vessel, an organ, or your whole entire body at one time, within a given period. We generally measure it as milliliters per minute, and it's relatively constant when you're not moving. It does, however, widely vary through individual organs. So some organs need a lot of blood flow, like your kidneys, your spleen, high blood flow through there, whereas others might need less. When your parasympathetic is taking over, you'll have more blood flow to your digestive organs, and then when you're not at rest, if you go into sympathetic, it'll decrease the amount of blood there. Animation you can click through. Blood pressure is the amount of force on the blood vessel walls as blood moves through. And we generally say that in terms of millimeters mercury. And when we want to measure our blood pressure, we measure it in our arteries of the systemic portion of your circulation. Why? Because those have the highest and most variable blood pressure. So if we want to know the most we can about our overall blood pressure, we want to look there. And remember that everything moves from where it's at higher pressure to lower. So as blood moves through our arteries to capillaries to veins, it's always going to be moving from higher to lower to lower to lower to lower blood pressure. If that wasn't the case, it wouldn't continue moving forward. It'll start out with a pressure that's quite high, and then by the time we're at the veins, like the inferior superior vena cava, coming back into the right atrium, we're at basically zero pressure. And resistance is opposition to flow. So we generally measure that as friction. While blood is in resistance, you'll have pressure, and then you'll see after the blood moves through, the resistance, blood pressure drops. So we're going to go through the three sources of resistance, where we get friction from. So first, blood viscosity, how thick it is, the total length of your blood vessels, and then the diameter of your blood vessels. The easiest of these to change is the blood vessel diameter. So we'll be able to do that to alter the resistance and the pressure in our vessels. So the factors that are relatively constant, your blood viscosity, how thick it is. So that's generally going to be mostly due to your hematocrit, the amount of your blood that's red blood cells. So if you've got a higher percentage of your blood that's red blood cells, a higher hematocrit, it's going to be thicker, stickier. That thicker, higher viscosity means that you're going to have more resistance. We can change this as our spleen breaks down red blood cells or as our red bone marrow makes new, but it's going to be a lot slower process than changing the blood vessel diameter. Whereas blood vessel length doesn't vary once we're done growing. And it makes sense that the longer the vessel, the greater the resistance that's encountered. So the farther things move, the more friction they're going to encounter. Blood vessel diameter is the greatest influence on resistance, and that's what we can change quickly. So think about if your blood vessel is dilated, you've got blood moving through, and then when you narrow it, you contract it, there's a lot of friction in there. There's a lot of resistance. So if we want to change our blood pressure, we can do it most quickly by altering the diameter of our blood vessels. If our blood pressure is too high, we can dilate them to decrease our blood pressure. As our blood moves through our vessels, the place where they encounter the most resistance is the arterioles before you get to the capillaries. So you'll see after blood moves through there, their pressure drops after that resistance. You'll also see large levels of resistance anywhere there's some sort of blockage. So if you have like a fatty plaque formed, That decreases the amount of space blood has to move through. There's going to be more red blood cells hitting each other, hitting the walls of that vessel. You've got more resistance. So now let's talk about measuring blood pressure. So your systemic blood pressure is the pressure generated by the left side of your heart pumping blood out to the body. And it's the highest as soon as it leaves the heart and the aorta, decreases the whole way through the pathway until you're back to right around 0 in the right atrium. So blood pressure, resistance, blood pressure drops, resistance, blood pressure drops, resistance, blood pressure drops. And we'll see the most resistance and then the highest drop of blood pressure in the arterioles. So here's a graph. You have systolic pressure. That's the blood pressure in your vessels when your ventricles are contracted. And diastolic, when the ventricles are relaxed. And you can see that there's the biggest difference between systolic and diastolic in those early vessels leading away from the heart. So aorta, arteries, highest pressure. And then you can see when we go through the arterioles, you have the greatest decrease in pressure, lots of resistance, and afterwards pressure drops. Then we move through the capillaries and start returning to the heart under very low pressure till we get back to the heart. The blood pressure in your arteries depends on two things. First, elasticity. That's how well they stretch, how well they recoil. And then as well, the volume of the blood pumped into them at any time. When you measure systolic pressure, that's the higher pressure. That's the pressure during ventricular contraction. Whereas diastolic is the lowest level of pressure. That's when the ventricles are relaxed. And when you feel your pulse on your wrist or in your neck, for example, you're feeling the difference in pressure between the two. So every time you feel the beat, that's going from diastolic pressure up to systolic and back down again. So if you've got a large difference, you feel a harder pulse. Venous pressure, you see very little change throughout your cardiac cycle. So if you look back at this graph, there's a big difference between systolic and diastolic in your early arteries, but once you get to the veins, you can't tell the difference. They're basically the same. And as you saw from the graph, there is very little difference in pressure from the venules down to the vena cava. It drops only about 50 millimeters mercury. And that's because we've had all this peripheral resistance, all this friction has decreased the pressure of our blood by the time we've gotten to this point. Okay, so if our veins have such low pressure, how do we get the blood back through? Well, first you have what's called the muscular pump, and we'll look at a picture of this next. Whenever you contract your skeletal muscle and you put pressure on the veins, that milks or forces blood upwards through valves, and then they can't flow backwards when the muscle relaxes. So every time you take a step, you're pushing a little bit of blood up. Respiratory pump is the same idea, but that's with your lungs and everything in your abdominal cavity. So that pressure forces blood forward towards the heart. And last, venoconstriction. However, because the veins have such large lumens and very little pressure, vasoconstriction of your veins doesn't do nearly as much as what it does in your arteries, where you've got smaller lumen, more pressure. So here's an example of a skeletal muscular pump. You can see as your muscles contract, they put pressure that forces blood upwards. It can't go down because the valve is there. And then as they relax, the blood that's up here can't go past the valve here. So we keep moving it up and up like a ladder until we get back to the heart. So how do we maintain normal blood pressure? Well, you have to have your heart, blood vessels, and kidneys working together. You'll also have your nervous system supervising as well. The main factors that influence our blood pressure are cardiac output. So remember cardiac output, how much blood is moving through a ventricle each minute. And that's based on your heart rate and the stroke volume. How much blood is moving through your ventricle each time it contracts. Peripheral resistance. Remember that's friction. And blood volume, how much blood volume there is. So if we want to change these things, we're going to change our cardiac output most easily by changing our heart rate. And remember, we just discussed the easiest way to change peripheral resistance is blood vessel diameter. And more long term, we can control blood volume by determining how much water we filter out or keep in our blood at the kidneys. So we'll look at the short-term neural and hormonal controls that help us control peripheral resistance, that blood vessel diameter, whether they vasoconstrict or vasodilate, and cardiac output, changing our heart rate. And then we'll look at the long-term renal regulation. Kidneys control the amount of blood volume. So first short-term mechanism, neural controls our nervous system. So these are going to operate via reflex arcs. So remember that's receptors taking in sensory information, sending it to our control center, the cardiovascular center in our medulla, the brainstem. That makes the decision of what to do and sends out a response to an effector that makes a change. Remember, effectors are either muscles or glands. So most often to do this, we'll use baroreceptors. A barometer is something that we use to measure pressure. Baroreceptors measure pressure in our body. And it can also come from chemoreceptors, chemical receptors, looking at the amount of different chemicals in our blood. And also our higher brain centers can sometimes override. If you remember back to the nervous system chapter, the brainstem has a lot of unconscious, involuntary, lower level functions, including the cardiovascular center that helps regulate heart rate, heart contraction, and blood vessel diameter. So baroreceptors monitor pressure. They're going to be located in areas that have high pressure. You're not going to find baroreceptors in our veins. That's not going to tell us anything. We want those receptors where there are areas of high arteries near our heart. These baroreceptors, something called mechanical receptors. They're mechanical. And basically when they get too stretched, that causes their gates to open and send an action potential. So they go off when they're under high pressure. So if the blood pressure is too high, baroreceptors, they send information to the cardiovascular centers. And what do we want to do to decrease blood pressure? We can decrease our cardiac output by decreasing our heart rate. We can also have the blood vessels dilate and decrease our peripheral resistance. Both of those resulting in decreased blood pressure. Good. So we decrease the peripheral resistance by dilating our blood vessels, so there's less pressure there. And then we decrease our cardiac output. We decrease our heart rate. We're pushing less blood through, there's less pressure. There's a nice animation to go along with this idea. We just looked at what we do if our blood pressure is too high. But what do we do if it's too... In that case, we look at chemoreceptors. These are chemical receptors, so they respond to chemicals in our blood. And they're going to respond if we have too much carbon dioxide, if we're not getting rid of enough at the lungs, or if we have too low of a pH, or we don't have enough oxygen, so we're not picking enough up. That means we need to pump blood around faster, get it through better. So what happens? We accelerate our heart rate, increasing our cardiac output. And we signal our vasomotor center to contract the smooth muscle of our blood vessels. Both cause the blood pressure to go up. So those are the general ways we increase or decrease our blood pressure. It can, however, be overridden by our higher brain centers. So in the next video, we'll start looking at that.